Okay, so in this class, we will talk about differential gene expression. In this class, we will talk about the introduction to gene expression. Then we will move to the postulate of genomic equivalence and the different levels of gene regulation. So let's start. Here you can see some readings, and that if you're interested, you can and you can find your textbook about this information. And let's start with the introduction to gene expression. Okay, so now imagine that you're in a city and it's during the night, and you find that some lights are on, and you look at other buildings and some lights are off. But imagine what happened if all the lights were on. Yeah, there will be a huge consumption of energy, and actually nobody will be able to sleep. And there is no need that all the rooms in a house are on. Yeah, and imagine the opposite case, that all the lights were off, were very unsafe and very insecure. So what you generally have is a intermediate stage where the lights that are needed are on and the lights that are not needed are off. Something like that happened with the gene expression. So in during gene expression, some genes need to be on and other ones need to be off to have normal development. Yeah. If all the genes are on, you will not have differentiation. If the inappropriate genes are on at the wrong time, you can have diseases like cancer. At the same time, if you have all the gene off, there will be not functional, there will be no function. The proteins are not going to be secreted at the right time. So yeah, so it's not about expressing the same gene, all the genes at the same time, or not expressing all the genes. It's expressing the right genes in the right location at the right time. Okay, so gene expression is fundamental for that. And let me show you another example with electronic devices. So for example, you need to have your laptop, your cell phone, the TV on, all the things you're going to only be able to watch one of them. Yeah, and you need to have the heating and the AC on during winter, during summer. You need to only have one of them at the because they are opposite. So the same thing happened with gene expression. If you have some genes on, all ones need to be off, and this is key for the gene expression. There is one exception that are called the housekeeping genes. Yeah, there are genes that are expressed consistently across tissues, and they are involved in the cellular maintenance across species, and you can find it here. Yeah, so those genes seems to be on most of the time. Now let's talk about the three postulates of genomic equivalence. Okay, so here you can read the first postulate, but the idea is that the DNA in all differentiated cells is identical. And this is exactly what happened in the ectoderm, the mesoderm, and the endoderm, and even in the germ cells. You will see that the DNA is identical in the germ cells. They will be diploid, and the other ones will be haploid. But the idea is that although they look different, they have the same DNA. It's just that they are expressed in different ways. The genes are expressed in different ways. The second postulate is that there are some genes that, as we said, they will be unused, yeah, in differentiated cells, but they are not destroyed or mutated. Yeah, so here you will have your RNA polymerase, and this will be able to express some genes, and other ones are not going to be expressed. So you just inhibit the expression of those genes, but they are ready in case that they need to be used. And the third postulate is that only a small percentage of the genome is expressed in each cell. Only a portion of the RNA is synthesized. Yeah. So this is exactly what happens. So if we have a specific RNA, maybe it's not going to be expressed in all these in most of the cells is only going to be expressed possibly in this one and possibly in this type of cells. And it's exactly the kind of cell of the that I told you about the electronic devices that maybe you just need to have one on and the rest off. And this is the big difference with the gene expression. Yeah. Now I want you to tell me about this idea, or more than idea as a question. Do these three postulate align or contradict what Dolly chip cloning experiments? Okay, so what do you think? Do you think that the experiments that they did with Dolly show that the DNA of all cells differentiate all the DNA of all differentiated cells is identical? Yeah, they took a mature cell, if you remember, and they put it in an egg. So to some degree it's true. And use genes in differentiated cells retain the potential to be expressed. That is true. And only a small percentage of the genomes are expressed. Yeah, so the in Dolly something like that happened. And what was the problem with Dolly? That Dolly died age older because there were some things in the DNA that we didn't know that they were already that it was not exactly identical. And why? Because the DNA to some degree can age too, and we will talk about that. We think that there were methylation groups or other things that show that the DNA can some degree of aging, and we need to repair them if we want to do this kind of experiments. But based on that, these three identical things are correct. And one of the first evidence of genomic equivalence is this picture that you see here. I don't know if you took genetics. This is a polytene chromosome. So what is a polytene? Polytene chromosome is a chromosome that has been copied multiple times. So you here, this is not one chromosome. This is hundreds of chromosomes. So one is here that is attached to another one here and attached to another one here and so on hundreds of times. And this showed that the information remained the same. Yeah. So why this giant chromosome appears? Because you need a lot of gene expression for the larvae to they need to produce like a glue to attach to the surface in Drosophila when they go to the pupal stage. So they produce in the salivary gland these huge chromosomes, and this is why and what you need so much of proteins, so they need to express a lot of them, and they create this adaptation. And this was a cool way to show that each chromosome is similar. You see all of them will have lines. Yeah, yeah, and these lines repeat and repeat. Yeah, so it showed that there is a genomic equivalence. And this was shown before. Why this was important? Because this was shown before we were able to sequence the DNA. So it was just through staining that they were finding that there was an evidence of the genomic equivalence. Now we're going to talk about the different levels of gene regulation. To talk about gene regulation, we need to go to the central dogma of biology, so to all your introductory biology courses, that the information in molecular biology goes in one direction. Yeah, that goes generally in this direction, from DNA to RNA, from RNA to proteins. And each one of these steps, they could be some degree of gene regulation, and we are going to cover some examples. The first example of gene regulation will be during transcription when you have an on and an off level. The second level, it could be during RNA processing where you can have alternative splicing. That I will talk in a second about alternative splicing. In the same time, at the RNA level, you can have different mechanisms to transport the messenger RNA, to give it stability, and to send it to the right location. So these are the three beginning mechanisms of regulation of the DNA regulation. Okay, let's talk about three more examples of gene regulation. So another one, it could be the translation, that the translation can be on and off. And we can also have post-transcriptional modifications. So the protein generally you can add them, put it in specific places. You can have interaction between proteins and the quaternary structures, and you can have an active gene product. A good example of gene regulation could be the different type of RNAs. So we generally talk about messenger RNA, ribosomal RNA, and transfer RNA. And this is the information that we generally talk in introductory biology. However, there are many more types of RNA. Yeah, there could be at least 11 different types of RNA that have different functions, especially in the regulation of the in the gene regulation. So let me show you some of the examples. So here you have the non-coding RNA that is responsible for a lot of the regulation. Yeah, so you can have it depending on the size, if they are small, if they are big. Some of them, for example, the microRNA is very famous, and it has been found involved in regulation. You can have uh, the interference RNA that is also very important in its mission, complementing the RNA, the messenger RNA, and again involving the gene regulation. But these are I just wanted to show you. And the trend is not that they will going to be reduced, but that they will be find more and more type of RNA that they will be in the regulation of, that they will be involved in the gene regulation. Another way to modulating the access to gene is through chromatin modifications. And here in this diagram, I know you have studied this in genetics, as you know you have studied this in possibly in molecular biology or in other courses, but this is a very important way in which developmental biology work. So let's go a small review. This diagram show you the key information that you need to remember. So the first one, if you remember, this is a nucleosome, and you see that each one of these three components is a histone, and there are 8. And in orange, you see, or cream color, you see the H1 histone. And in number 4, you see the DNA wrap that it gives 2 turns. And number 5, you see the linker DNA. Okay, and approximately half of the weight of the DNA is due to the proteins, the histone proteins, that as a whole are called nucleosome. As a reminder, there are, as I said in the previous slide, there are 8 proteins, 8 histones, and you can see there is H1 is the one that is linking the 2 nucleosome. But here you have H2A, H3, H2B, and H4. And here you will see again the same copies. And here you will see the tails of each one of these histones that are very important, because it's the place that is going to allow the repression or is going to allow the activation of the gene expression. So here again you can see the chromatin that is very well packed, that it makes that 6 feet of DNA look in like 6-inch. And here you see the nucleosome, and you see the space and the tails. Yeah, and now the key information is how these chromatin modifications allow or prevent the DNA to be transcribed. And the key information there is I will show you in the next slide. Okay, so here you can see 2 diagrams. Yeah, the one on the top shows the condensed DNA, and the one in the bottom shows the and contains DNA. And which one do you think that the DNA polymerase is going to attach? So of course, the one on the top is not the DNA polymerase doesn't have a space to attach, while the one in the bottom, the one that is acetylated, it have place to attach. So in those one is the one that uh, the gene regulation will be activated. Well, in the one that is methylated, the DNA methylation will not allow the attachment of the polymerase. If you remember, we were talking about Dolly a few slides before, and I told you that the story was more complicated, that the three postulates are not violated, but we didn't know that the old cell that they took already have some methylation and some acetylation. So to bring completely the cells back, the DNA back, we need to also play with this field that is the chromatin modifications. That is a key way to regulate genes. So again, the same schematic is here, but now we're going to focus on here. That is where the methyl groups are located, and each one will have a specific function. Yeah, it will regulate a specific function in the genes. So what is histone methylation? Histone methylation is the addition of a methyl group by an enzyme called methyltransferases. Yeah, and to add in a methyl group will repress the transcription, as I showed before, and getting rid of those methyl groups is expected to allow the transcription to start again. So here I just wanted to show you an example of what is the nomenclature of the methyl groups. So here you can see that this one is called H3K27 trimethylation. So I color coded this for you, and here in green you will see that the K27 come from the lysine at position 27. The red one that you can see here, H3, main the histone, and the trimethylation group is located here. This is one, this will be a second, and this is the third one. I think this is something that maybe I'm going to ask you this in the test, but in case that one of you end up working in methylation, this is something that maybe later you will use it. In addition to all this regulation that I just talked to you during this class, there is another one that is simpler that maybe you have here, that is the alternative splicing. There's a way, in another way, that during development they regulate, they increase the gene regulation. So here you can see a messenger RNA, and let's start with this one, and that has 1, 2, 3, 4, and 5 exons, and in between you find in gray all the introns. And during alternative splicing, you can get this messenger RNA that is only have 1, 3, and 5. But you can also, another example will be this one below, that it has 1, 2, 4, and 5. And you can get another one. You can get 1 and 2. Yeah, you can also get 1 and 5. You can get 1, 3, 4, and etc. So in this way, you have a combinatorial power to produce as many sequence, and in that way you can have more regulation in the gene, and some genes will be expressing in specific tissues and at specific times. Okay, in this class we talk about differential gene expression during development, and we start with the introduction of gene expression. Remember that I told you that not all the genes can be on and off at the same time, and there are only few exceptions that are genes that called housekeeping genes that are generally on. And we talk about the three postulates of genomic equivalence, and that all of them when we have these clonic experiments, we can see that they still work. And I show you that there are different levels of gene regulation. And I talk about alternative splicing type of RNAs. We also talk about chromatin modification. And these are just some examples. There are more that every day emerge
that are very important, and during the course we will talk about more in following classes. Okay, if you hope that this it was clear, and if you have any questions, just let me know. Have a good day and bye.